Hello, my name is Emily, and today I will be describing the structure of a neuron and the functions that go on within it. I have created a neuron out of food products that I have found in my house. To begin, I'm going to explain the different structures within a neuron that I have constructed out of food. To start from left to right, this structure on this side is called a dendrite. It receives information and sends it towards body cells. It is composed of spaghetti and parsley. Next is the cell body, which is a part of the dendrite. It is the orange right now. It is kind of the control center of the dendrite. Next is the axon hillock, which is the connection between the cell body, the axon, and the dendrites. At each end of the skewer that is running between the carrots here, there's a hook at each end, and that represents my axon hillock. Next is an axon, which is a canal that passes messages along to other neurons and muscles and glands. It is composed of a skewer that is running through the carrots. The next part of the neuron are the nodes of Vannier, which are the gaps between each compartment of the myelin sheath. The myelin sheath is composed of carrots right now, and between each of them is a node of Vannier, which is the space between the compartments. The myelin sheath is a selectively permeable membrane that allows positive and negative ions to enter the axon. It is carrots right now in my example. Next is the terminal branches. They form a junction with other cells. It is currently grape stems. They form junctions with other cells. On the inside of the myelin sheath, there is negative ions, and they are represented by rice. I don't know if you can see those, but they are there. And also cotton tops, like cotton swab tops, represent the positive ions that are currently resting outside of the myelin sheath. Next, I'm going to describe the different processes that go on within the neuron. First is resting potential, which is the state of those positive ions, the Q-tip tops, and the negative ions, which is rice, being inside and out of the myelin sheath. Next is what's called depolarization at the dendrites, which is where sodium and potassium ion channels open, so that the channels within the axon open and allow for sodium, potassium, positive and negative ions to travel through. Next is the threshold. When a neuron reaches the threshold, it activates other gates within the dendrite. So when the neuron reaches the highest point, it activates the gates within this dendrite to cause it to function. Next is depolarization at the axon. The first bit of the axon gate opens, and positively charged ions, which are the Q-tip tops, enter that section through the membrane. This depolarizes that section. So one or all of the positive ions would flood into the first section of the neuron in the axon, and it would depolarize this section. It opens the next gate to the next section, which would be the next carrot in my example. The neuron then pumps the positive ions back into the membrane. So they enter through one, flood all of the capsules, and then flow back out. Next is action potential in electric form in axons. The action potential floods each segment and causes it to be recharged. That's when they all enter the segments and then flow back out at the end. The release of neurotransmitters is when the action potential reaches the terminals at the end of the axon's end, and it triggers the release of chemical messages called neurotransmitters. They jump across the synaptic gap, which would be the gap between this particular neuron and the next one that would be receiving its messages, and they bind themselves to receptors on the receiving neuron. The neurotransmitter unlocks a gate and allows electrically charged atoms to flow in, exciting or inhibiting the neuron, like pushing the gas pedal or pushing on the brake. Reputake is when the sending neuron retracts the excess transmitters that were not used to excite or inhibit the receiving neuron. This process is called reputake. I forgot that when the neuron reaching the threshold is when excitatory signals minus the inhibitory signals trigger a reaction called action potential. This travels down the axon and into thousands of junctions with other neurons and passes the body's muscles. So that's when it's traveling through the axon and causes action potential. So that's my video. I hope it was informational.